Hi everyone, I'm Sandhya Vasudevan, and today I will be talking about the role of minor tissue absorbers in distinguishing benign and malignant breast lesions using broadband diffuse optical imaging. Mammography is the most common screening technique for breast cancer. On this slide, you can see two mammograms. One is taken on a fatty breast and one is taken on a dense breast. Now, in the fatty breast, the mammogram is able to identify even a small cancer. However, despite of there being a relatively larger cancer in the dense breast, the mammogram is unable to identify this. This tells us that mammogram sensitivity is limited in dense breast tissue, which is predominantly found in younger women. In such cases, adding ultrasound screening does increase the number of cancers detected. However, it is still hard to identify what the detected lesion is, whether the detected lesion is benign or malignant. Since that question remains unanswered most of the time, this leads to an increased number of unnecessary biopsies. So, unnecessary biopsies are as high as 71% in the United States, and this translates to an annual cost of $2.18 billion. Keep in mind that these are biopsy procedures that could have been avoided, and they also end up causing a lot of emotional and physical distress to the patients going through these procedures. So there is a clinical need for an imaging technique that can better discriminate benign and malignant lesions. An imaging technique that has shown some significant potential in this area, which is what we will be talking about today, is diffuse optical imaging, also known as DOI. In diffuse optical imaging, visible and near-infrared light is used to macroscopically image biological tissue. This is a non-invasive imaging technique which provides clinical diagnosis of the underlying tissue structure and function. Here is a picture of the handheld probe of our diffuse optical instrument. Now, how we use this probe is that we keep it in contact with the tissue surface and we manually move this probe along a rectangular grid pattern to acquire measurements. Our measurements are finally converted to 2D maps of tissue optical properties such as tissue absorption and reduced scattering. Now let's take a look at the role of major tissue absorbers when it comes to differentiating benign and malignant lesions. So here we have four major tissue absorber maps overlaid on a breast mannequin, which is oxyhemoglobin, deoxyhemoglobin, water, and fat. And this mannequin over here has a tumor-containing breast and a normal breast. Now, many DOI studies have investigated the role of each of these four biomarkers in differentiating benign and malignant lesions. However, most DOI approaches require normalization of these chromophore values in the lesion to the normal tissue or the healthy counterpart, since these are major tissue absorbers and they are subjected to high variability from one patient to the other. Also, none of these chromophores are unique to the tumor. They are also found in perfectly normal healthy tissue, and some benign lesions have also shown to express similar contrast. Some DOI studies have also extracted a spectral biomarker that is unique to the tumor, and these spectral biomarkers are known as specific tumor components or STC spectra. Now, here is one such published results where you're looking at the STC spectra between plotted out for 11 benign and 10 malignant lesions. And as you can see from the plot itself, there's a lot of contrast between the spectra for benign and malignant lesions. So these UI studies have established clinical significance of these interesting signatures in differentiating between these two lesion categories. However, the biochemical origin of the spectra remains unknown. Now, the spectra is a misfit of the basis chromophores, which is oxy, deoxyhemoglobin, water, and fat, to the difference between the normal and tumor absorption. So, the spectra over here could be indicating the presence of other minor absorbers that is not covered by these four major absorbers. So, what we did in our study is to observe this patient data set to uh, investigate the role of minor absorbers such as collagen and methemoglobin in differentiating benign and malignant lesions. So we performed a retrospective clinical study involving 30 lesions, 12 benign and 18 malignant lesions from 28 subjects. We reprocessed the raw data, which included frequency domain data and continuous wave data, and determined broadband absorption and reduced scattering in a wavelength range of 640 to 1000 nanometers. Now, from the broadband absorption, we extracted all the chromophore values of interest. So we in total looked at six chromophores. The extinction spectra of all those six chromophores are plotted out over here, which includes the four major absorbers as well as two minor absorbers, collagen and methemoglobin. Now, from the broadband absorption, we extracted scattering parameters using this inverse power law. So let's take a look at what are the variables of interest involved in the study. So we look at all the variables in two different forms. So one is a normalized form and one is the absolute form. Now, in the normalized form, the variable values are listed out as ratio of the lesion to the normal tissue in the contralateral breast. Whereas when you look at these values in an absolute form, you're observing these values only within the lesion. It has no reference to the normal tissue. What we did is we performed a statistical analysis on all of these DOI variables. The statistical test that we performed is a Wilcoxon-Ransom test, and statistical significance was assumed for the variable if the p-value falls below 0.0063. Now, this threshold 0.0063 has been corrected for multiplicity using the Bonferroni method, which is why it is such a small threshold. And on this slide, you're looking at a box plot of all the normalized variables included in the study. Now, what a box plot is, it shows the distribution of each of these variables between the two lesion categories, the benign and the malignant category. And the p-value of each of those variables are listed above over here. Those p-values which fall below the threshold, which are significant, are written out in blue with an asterisk sign on top of it. So in all of these normalized variables, we found that four variables showed significance in separating benign and malignant lesions, and those were oxyhemoglobin, deoxyhemoglobin, water, and methemoglobin. Let's look at how the absolute variables performed in the statistical test. So here is again a box plot representation of all absolute variables with their p-values written out. Now this gave us an interesting result. Among all the absolute variables that we looked at in this study, we found only one variable to be significant in its absolute form, and that was methemoglobin. Now, this points out that methemoglobin is a unique absolute biomarker of breast malignancy that does not require any normalization to a healthy tissue. Now, in this slide, we are just looking at all of the plots that we saw in the previous two slides together. So we are looking at the normalized and the absolute variables together. And here I have highlighted all of the significant variables in here. So altogether, there were 5 variables that showed significance. 4 of them were in the normalized form and only one was significant in its absolute form. If you look at each of their p-values, you can see that absolute methemoglobin has the lowest p-value of which is 0.0002. So this shows that absolute methemoglobin is the most significant parameter amongst all the parameters, normalized plus absolute parameters included in the study. Now let's try to understand how this significance of methemoglobin is biologically justified. Now, some biological studies have shown that increased methemoglobin has been shown to correlate with higher proliferation in cancer cells. Also, hemoglobin oxidized to form methemoglobin in conditions in the tissue such as neovascularization and hemorrhage or leaky blood vessels, which is usually found in malignant conditions that can lead to uncontrolled tumor growth. Also, tumor cell-derived nitric oxide, which is produced in conditions like inflammation of the tissue, hypoxia, or metabolic stress in the tissue, could also result in conversion of hemoglobin to form methemoglobin. I would like to emphasize over here that this is the first time that methemoglobin is being examined in breast lesions. and we have identified that methemoglobin has significant potential in non-invasive lesion characterization. Now, to conclude, we performed a quantitative broadband diffuse optical imaging to characterize 18 malignant and 12 benign breast lesions. We investigated a total of 8 DOI variables, both in the normalized and the absolute form, amongst which 5 variables were shown to be significant in separating benign and malignant lesions. 4 of these were in the normalized form, which is oxyhemoglobin, deoxyhemoglobin, water, and methemoglobin, and only one was significant in the absolute form, which is methemoglobin. Methemoglobin concentration had the lowest p-value, indicating that this is the most significant parameter that is able to differentiate between the lesion types, suggesting it to be an important optical biomarker that needs more research. This is the first time that methemoglobin is being examined in breast lesions, and we believe that it will have a significant effect in characterizing lesions. Future studies should include more investigation of methemoglobin, particularly among different lesion types, such as cysts, fibroadenomas, and other benign lesion types. With that, I would like to conclude my talk and acknowledge all the funding resources that made this research possible. Thank you for your interest in the topic.